Hello boys, today we are going to discuss about the structure of the cell. So we all know about the cells and we have studied about the cells in basic in our previous classes as well. And today we are going to elaborate it further as a part of our syllabus of and a chapter of class 9 as well. So what is a cell? How we are going to describe a cell? So basically the cell is considered to be the structural and functional unit of all the living organisms. Whether it is a plant, whether it is an animal, whether it is a cow, whether it is a whale or whatever that we see around us, these are all made up of cells. But in addition to that, we should also know that there are certain exceptions to that as well. And we all know that in science, especially the biological science, and the biology is also considered to be the science of exceptions because you can find large number of exceptions in that as well. So there are certain things, the certain organisms which we call as viruses, which are exception to the cells. For example, the viruses can show the characteristics of living as well as non-living. For example, the viruses can be dried and stored in the form of powder as well. On the other hand, when the viruses are there inside a host cell, a living cell, then they show all the characteristic features of a living organisms. And these days, we all are suffering with the viruses around us. The COVID virus, for example, the influenza virus around us, the herpes virus. So these are certain examples of viruses. We will discuss about the viruses in our next lecture. So now we are going to discuss about the diversity in the structure of the cells. So cells are around us are seen or observed in various shapes and sizes. Okay, so first of all we will discuss about the size of the cells. Some of the cells are so small that they are observed with the microscope even with great difficulty. For example, the mycoplasmas. These are actually cells, but these are very small, so it is very difficult for us to observe them even by using the microscope. But some of the cells are so large and so big that they can be easily observed with the help of naked eye even. For example, if we talk about the plant cell, the largest among the plant cells is known as the acetabilia. It appears like a small plant, but in fact it is a cell. Similarly, in the case of animals, the largest among the cells is that of ostrich egg. Ostrich egg is sufficiently large and can be seen with the naked eye. Remember, the ostrich egg is a cell till it is unfertilized. Likewise, the human body also has a large cell. This large cell is known as the nerve cell. So nerve cell in the case of human body is the largest of all. It can extend up to a length of 1 meter in certain cases. But there was a bottleneck in observing the cells. Why? Because the cells in normal case are infinitely small in size, or you can say very small in size. So we need specialized instruments to observe them. For this, we need some microscopes. But earlier, the microscopes were not there. But for the first time, the microscope was invented by not a biologist, but he was a small-time merchant. His name was Antony von Leeuwenhoek. So Antony von Leeuwenhoek for the first time made a simple microscope in which he used a lens which he grounded himself and through which he observed small organisms in dental scraping and in rainwater. So since he was not a biologist, not a man of biology, so that is why he called them as animalcules. So the Antony von Leeuwenhoek's microscope was of very simple type. It had good resolution, but it was of very simple type. Later on, Robert Hooke invented a microscope which used a system of lenses and filters. It was known as compound microscope. And compound microscope was able to provide with more fidelity and more clear resolution, and the images were crisp sharp. So with the help of the Robert Hooke's microscope, the structure of the cell was very easy to learn and understand and observe as well. So now we are going to discuss about some more advancements in microscopy. So the next microscope is known as the electron microscope. What is the difference between electron microscope and a light microscope? So if we know a general idea about the electromagnetic spectrum, we know that electromagnetic spectrum lies between 470 nanometer to 780 nanometer. So anything which is above 780 nanometer is invisible for us, and anything that is below 470 nanometer is invisible for us, because these will never cast any image on our retina. So electron microscope uses an entirely different strategy. It uses the part of a spectrum which is lesser than our visible spectrum, because the wavelengths of higher spectrum are harmful for us. So it uses the visible spectrum, it does not use the visible spectrum. In fact, it is using the visible spectrum, which is the electron microscope uses the spectrum which is below the range of visible spectrum. For example, the wavelength used by electron microscope lies in the range of 65 nanometer to 100 nanometer. So here the source of illumination in the case of electron microscope is not the light, but in fact the beam of electrons. So who invented the electron microscope? There were two Russian workers, Noll and Ruska, who for the first time designed an electron microscope. There are two types of microscopes these days which are utilized worldwide. These are the scanning electron microscope, which scans the surface and provides a three-dimensional image. And second one is known as the transmission electron microscope, in which the electron beam travels through the fixed specimen and provides a image on the computer screen or a projector screen. So there are certain limitations with the electron microscope as well. For example, in the case of electron microscope, since you are using a wavelength which is far less than visible spectrum, so don't expect colors here, you will obtain only monochrome images. But the resolution and fidelity in the case of electron microscopes is relatively very high in comparison to the normal optical microscope or the normal compound microscope. Now, let us discuss something about the structure of the cells once more. We have discussed about how the cells are observed. Now we will discuss about how the cell theory was formulated. What was the need of the cell theory? Because people around the world had different concepts regarding the cell theory. So it was needed that we should formulate a cell theory which should generalize all the observations. So what were the observations? Let us discuss about them as well. So the cell theory was propounded by two German workers, Matthias Schleiden and Theodor Schwann. So this cell theory says that the cells are essential components, or the cell is the structural and functional unit of life. Whether the plant or an animal, these are all made up of cells. Okay? Energy flow occurs always inside the cell. You cannot expect a cell to teleport energy from one place to another place which has a cell. The cells, the flow of energy between the cells, inside the cells, always takes place with the help of the cellular activities, and inside the cell and between the cells only, without the aid of any external medium. Or to be very precise, the required energy for the metabolic processes, or the energy required to carry out the processes for the cell, are generated by the cell itself. Now the third point. The third point came a little bit later, because till the discovery of the cells, the workers around the world were not clear about the evolution of life on Earth. There were many theories. So there was a worker known as Adolf Rudolf Virchow. He said that the cells arise from pre-existing cells, means to say that you cannot simply mix the ingredients together and expect that you will generate a cell spontaneously. Or to be very precise, again we can say the de novo formation of the cells is technically impossible. Even if you get all the ingredients together, you cannot synthesize a cell without a living system. That means a daughter cell is always produced by a parent cell. So cells do form or do follow a parental line or a genetic line. The cells arise from pre-existing cells. So with this, we are going to finish off this lecture, and hope you must have enjoyed it. go through the lecture again. If you find any difficulty, feel free to discuss with us. Thanks a lot.